Hello everyone, and welcome to this audio podcast. As part of the Storm Mode Isomer. This audio podcast as part of Mayhem and Twister. This audio podcast as part of the Milliplex, and this audio podcast as part of the Storm and the Storm Robotics production. So continuing in on the fantastic works by the fantastic works in on works by science student one of the science students from an academic institution in the southwest UK. One of the science students, Heidi Stefan, who actually did some fantastic works or looked at some fantastic works on looking at Oxford nanopore sequencing technology involvement in the genomic domain and its impact in the biomedical science field. There is now looking at now in this audio podcast there will be a look at some of the advantages and disadvantages of nanopore sequencing technology. So in around some of the advantages, nanopore sequencing software and devices are sort of are sort of are sort of sort of more sort of powerful enough in the sequencing discipline, allowing the sort of construction and use of long base reads from quantity DNA and RNA rapidly. That is to say, listeners and viewers, that in amongst nanopore sequencing technology, that for example whole DNA or whole RNA is not necessary, is not a necessity. So therefore, only small amounts of RNA or DNA can be used or can be utilized or are utilized in the analysis for an analysis around a particular sort of causations of particular conditions, etc. So uh, in this additionally to highlight that that additionally to highlight that that uh, that the technology that nanopore sequencing technology can sort of execute a sort of can execute genome identification, beating sort of other sort of technologies, other technologies like Illumina, which has sort of a less genome accuracy. The potential of nanopore sequencing also allows exponential research into the genomic areas, allowing more information to be collated on entire genome specific specific molecules, genetic disease identification, and research in restricted environs highlight and research and research that is restricted in that can be restricted in particular areas or particular sort of scientific and scientific specialties. Um, so in and around this, additionally, some overview in around some of the some in around some of the disadvantages as well. So nanopore sequencing is a sort of a third generation technique, opening the genomic research potential. However, it still has disadvantages which need to be considered, such as specific handling requirements and error rates in long reads. An Oxford nanopore sequencing technology can potentially further develop can help can be further developed to reduce disadvantages, opening more opportunities for genomic investigations to improve to improve patient care and outcomes in amongst the biomedical science field. But some of the disadvantages and advantages are in around, for example, where in around both, advantages. I mean, the advantage, for example, is around the live data analysis, in around the portability, some of the inner aspects, and the vast options of library preparations, in around the scalability, the read lengths, the direct and direct analyses, in around the disadvantages is around the precise handling requirements, some of the aspects of error rates, software complexity, and software complexity. So that is a sort of a quick sort of snapshot in around the overview of advantages and disadvantages of in around nanopore sequencing technology. Um, that draws this audio podcast to a close. This audio podcast is part of Storm Mode and Isomer. This audio podcast is part of the Mayhem and Twister. This audio podcast is part of the Storm and Storm Lobostics production.